Cholesterol is all about the cell membrane of animal cells. Let's look at the structure of cholesterol. Cholesterol is a pretty regular-looking aliphatic fatty substance, but rather than long fatty acid chains, it has these four rings, and it has this double bond. So it is fixed in structure and fairly rigid. Cholesterol includes a long chain, but there's a hydroxyl on the end. The role of cholesterol in membrane structure is related to these structural details and solubility questions. Fats, including cholesterol, are not soluble in water. They are referred to as nonpolar. So this part is nonpolar, but the hydroxyl is polar and soluble in water. Let's look at the structure of membranes, starting with this. This is a triacylglyceride. This is a typical fat, as found in both animal and plants. This is the fatty acid side, long straight chain. They come in even numbers and include double bonds. That is, they may be polyunsaturated. The double bonds are cis. It's nonpolar and not soluble in water. This is a typical lipid. But look at this side. This is the glycerol end. It's polar and soluble in water. If fat is mixed with water, it spontaneously forms these little balls such that the polar ends are out toward the water and the fatty ends are together in the middle. This gives us an image of how fats are moved through the blood. The lipids are packaged into these little bundles surrounded by hydrophilic proteins, which is the LDL-HDL language. Animal cell membranes are similar. The fatty acids are in the middle, and the polar part is toward the outside of the cell or toward the cytosol, the inside of the cell. So how does cholesterol fit into this? Cholesterol. Let's represent it this way. The cholesterol molecule is incorporated into the cell membrane and stiffens the membrane. The hydrophilic part is outside, and the hydrophobic part is inside with the fatty acid chains. Cholesterol serves to fine-tune the fluidity of the cell membrane. All animal cells use cholesterol in this way. Cholesterol is made in every animal cell. There are 30 steps or so, each with an enzyme, a gene product, each controlled by a transcription factor, also a gene product. From a genetic and metabolic standpoint, it's a pretty big deal. Every animal cell has this machinery. Plants have a cell wall made out of cellulose as structural material. It's a carbohydrate. Plants have no use for cholesterol and never make it. Cholesterol is never used as an energy source, as are other fats, and animal metabolism has no mechanism for degrading excess cholesterol. It must be eliminated. Let's look at the whole organism. Every cell uses cholesterol and makes what it needs. Complications are that cells do not degrade it. There's a pathway for the degradation of every other category of molecules, but not cholesterol. And animals that eat meat ingest cholesterol with their food. On an organ level, the big player is the liver. The liver makes cholesterol, which can be added to the blood as needed, and excretes cholesterol, or pulls cholesterol out of the blood. The liver makes the extra cholesterol into bile, puts it into the gallbladder, which puts it into the intestine for elimination with the feces of the animal. Cholesterol can be reabsorbed from the intestine. A diet that's high in fiber helps eliminate excess cholesterol with the feces. The specifics of this depend upon the diet of the animal. Plants do not have any cholesterol. Absolute vegetarians, herbivores, may not even have a gallbladder. Mice and deer don't have a gallbladder. Processing extra ingested cholesterol will be very much part of the metabolism of a carnivore such as a dog or a cat. Why do humans have trouble with cholesterol? Maybe our long history as omnivores leans more toward being vegetarian, and our systems are not well evolved for processing large amounts of cholesterol.